Hi, welcome to another episode of Catch This. My name is Trudy Ray, and today I would like to spend some time talking about one of the SARS-CoV-2 vaccines, or at least a couple of them. I'm referring to the Moderna and Pfizer vaccines because they use the same technology. These vaccines are called mRNA vaccines because they consist of an mRNA, or messenger RNA molecule, that is encapsulated in a lipid nanoparticle which protects the mRNA from degradation and ensures proper delivery into cells. The mRNA codes for the SARS-CoV-2 spike protein, which has been the primary antigenic choice for a number of SARS-CoV-2 vaccine candidates. The reason for this is because the spike protein mediates binding of the virus to the ACE2 host cell receptor via its receptor binding domain, and it also mediates fusion of the viral particle with the host cell membrane via its fusion domain. Both of these domains are highly immunogenic and are targeted by neutralizing antibodies. Neutralizing antibodies not only bind viral antigens, but they also inactivate the virus and prevent infection of new cells. So how exactly does the vaccine work? Before I explain that, I want to briefly describe what is known as the central genetic dogma. Generally, our human DNA is transcribed into RNA, which is then translated into protein. The Pfizer and Moderna mRNA molecule is a messenger RNA, meaning that it doesn't require any additional processing and is immediately translated into a protein, the SARS-CoV-2 spike protein, which is then recognized by our immune system as foreign. Our immune system then produces antibodies against this protein. When a person receives this vaccine, the mRNA enters that person's cells and is translated by the host protein synthesis machinery into the SARS-CoV-2 spike protein. So which cells does the mRNA end up in? That depends. It can enter some local muscle cells, and the vaccine mRNA is then translated into protein inside those cells, and that protein is recognized by immune cells as foreign, ultimately leading to production of antibodies and T cells. Or it can enter local dendritic cells, which are known as professional antigen-presenting cells, because as soon as the vaccine mRNA is translated into protein inside these cells, that protein will be recognized as foreign, and the dendritic cells will head to the lymph node where they will present the spike antigen on their surface to T cells of the immune system. Or the vaccine mRNA can simply enter local muscle cells, which immediately die due to the trauma caused by the injection. However, those dead muscle cells will then be swallowed by dendritic cells, and the mRNA will follow a similar path as I just described with the dendritic cells. RNA-based vaccines are a relatively new technology, meaning that there haven't been any licensed mRNA vaccines yet. In fact, it wasn't until last week that the FDA authorized use of Pfizer's vaccine under an EUA, which stands for Emergency Use Authorization. This means that under certain circumstances, the FDA may allow the use of unapproved medical products or unapproved uses of approved medical products in an emergency to diagnose, treat, or prevent serious or life-threatening diseases or conditions when certain criteria have been met, including when there are no adequate, approved, and available alternatives. And we happen to be in that situation right now. I have had many people ask me if this vaccine is safe. It is considered safer than traditional vaccines because it doesn't contain infectious virus. And although there have been no licensed mRNA vaccines prior to now, mRNA vaccines have been tested in humans before for at least 4 infectious diseases: rabies, influenza, cytomegalovirus infection, and Zika virus infection. mRNA vaccines are particularly suitable for emerging pathogens because they can be produced more quickly and cost-effectively than traditional vaccines. Some people have been concerned because of the alleged possibility that this mRNA could integrate into our DNA and remain in our DNA forever, kind of like HIV does. This cannot happen, and I want to briefly explain why. HIV is an RNA virus that goes against the central genetic dogma. Its genome consists of RNA, which is reverse transcribed into DNA in the cytoplasm of our cells after it enters our cells, using an enzyme called reverse transcriptase. That reverse transcribed DNA then enters our nucleus, where it is integrated into our DNA using an enzyme called integrase. Both reverse transcriptase and integrase are enzymes that are encoded by HIV. The SARS-CoV-2 mRNA and the protein that are produced from this mRNA both only stick around in the cell for a short period of time. The mRNA sticks around long enough so that it becomes translated, and the protein sticks around long enough so that it can induce production of antibodies. Then after a while, they degrade. Unlike with HIV, there is no reverse transcriptase to make DNA from the vaccine mRNA. Since there is no resulting DNA, it does not go to the nucleus, and even if this did happen, there is no integrase enzyme to integrate the DNA into our genome. Even so, some people have been concerned that the foreign DNA can still insert into our genome using line 1 elements. Line 1 stands for long interspersed nuclear element 1. Lines are regions in our genome that can transpose or move themselves to different places in our genome, and certain portions of line 1 encode a protein that has reverse transcriptase activity. So the concern is that the vaccine mRNA could hijack these reverse transcriptase and insertion functions of line 1 elements and thus be inserted into our DNA. However, there are plenty of data showing that line 1s preferentially move their own genome, and this is just one example of a published paper showing that. There is no reason to think that a foreign novel RNA would have an advantage over what is already present in the genome and be reverse transcribed and inserted by these line 1 elements. Long story short, any concerns about the vaccine changing our DNA or becoming a permanent part of our genome are completely unfounded. That's it for today. Thanks again for listening. My name is Trudy Ray. I'll see you next time on Catch This.